Hello, everybody. My name is Bruna Moreira. I am a PhD student at the University of Melbourne in Australia. This poster today is to talk about a computational tool called CSM-epitope, which predicts linear B-cell epitopes using graph-based signatures and interpretable machine learning algorithm. I will start this presentation with a background, answering what is epitope and why we are predicting them. Moving to the methods used to build this tool, also sharing our preliminary results and the conclusions we have reached so far. So to start with the background, what is an epitope? We can't understand epitopes if we do not talk about how our immune system works. And basically, every time a threat invades our body, our immune system will take action, and it has some layers of defense. The most sophisticated one is responsible to produce and reproduce hundreds of thousands of antibodies that in turn will act to neutralize and also kill all these threats. We can also call these threats as antigen, but there is a specific mechanism between antibody and antigen, in where the antibody will not bind to the entire antigen molecule. Instead, it will bind to a specific part of its surface. It's called epitopes. So in other words, epitopes are small stretches on the antigen surface that are responsible to trigger a powerful immune reaction in the antibody side. And after that, the host, the body, will develop a long-term protection against this antigen. So CSM-epitope is a machine learning predictor that identifies linear B-cell epitopes with the aim of reducing the time and also the cost of the experimental approaches within this field that are extremely laborious and expensive. We start our model collecting some benchmark datasets to fairly compare our model with previous state-of-the-art models. And then we newly evaluate a graph-based signature, which is a mathematical modeling to map those residues pairs as nodes of a graph, and the interaction amid these pairs as edges. And building this network, we can help to identify between the differences between epitope and non-epitope given an input sequence. Then we selected a glass box model to have a high accuracy performance, but also giving us biological insights of how and why the model is making those decisions. And to an end, we will deliver it as a web server interface. So moving to the results, we are releasing here two new things. One is a large-scale curated dataset composed of only experimentally proved epitopes and non-epitopes. This dataset is composed of a bit more than 200,000 data points, which is 6 times larger than the current dataset, the larger one. We are also evaluating a new feature, and the graph-based signature was able to help identify differences between epitope and non-epitope, given some labeling information based on physicochemical properties and also scale propensity information. We can see in this table on the left, in a tenfold cross-validation technique, that CSM-epitope outperformed the previous methods, reaching an MCC of 0.72 against the current state-of-the-art performance epitope VAC that reached an MCC of 0.62 in the same dataset and scenario. On the right part, we have a chart in blind test comparison using 3 different datasets. We can see in the red color representation that CSM-epitope also outperformed the majority of them, except for one case that we are slightly worse than epitope VAC. And on the left corner, at the bottom of the poster, we have an illustration that is a feature contribution where we can see how the model is making the decision considering only that specific feature. but we will have this illustration for each feature applied in the model. This is just an example to see how the model is using the endogenicity scale, which is a very important feature for us, to distinguish between the two classes. And we have a very clear decision point around feature value equal 5, where model will score the higher probability of being an epitope if it falls above this value. On the contrary, if the given input sequence has a feature endogenicity scale in a value below 5, the lower the probability of being an epitope. And we can do this step for each feature to analyze the contribution of each one to the model final decision. So as a conclusion, CSM-epitope was capable to generalize between different data sets. And using the interpretability outcomes in visual plots illustration, we are able to understand the constraints and also some trends in every single benchmark dataset that we have the chance to evaluate here. And to sum up, we are releasing a new large-scale dataset with an updated information that covers a diverse number of different organisms. That's all for today. Thank you very much for your time, and feel free to contact us or reach us with any doubt or questions.